Hello, welcome to the lecture, and I'm Thomas Ming-Shui Chang. I've been working in artificial cell for more than 60 years. It's a pleasure to briefly summarize this area for you. First, let's ask a question. This is an example of the possible uses. I'll let you look at it. Microdialysis, drug delivery, blood substitute, biotherapeutic, vaccine, cancer therapy, immunotherapy, enzyme gene therapy, cell therapy, biotechnology, nanotechnology, regenerative medicine, agriculture industry, nanocomputer, nanorobotic, nanosensors. Let's look at cell and stem cell therapy and regenerative medicine. Cell inside artificial cell is excluded from outside immunological cells or antibody and therefore can avoid rejection. Artificial cell containing cell now has four major groups of exploration: cells and tissue from the body, genetic engineering cells, microorganisms, stem cell. For instance, you can put hepatocyte liver cell inside artificial cell, and it's prevented from rejection. These are red hepatocyte in mice quickly free-form rejected. If it's in artificial cell, it's not rejected. This is in artificial cell containing hepatocyte in Gunnarate congenital hyperbilirubinemia. The level of bilirubin in the animal increased. When you inject the artificial cell, it slowly decreased back to normal. You can also put this using liver cell. The problem is that after injection, it only works for 1 year. After 1 year, the artificial cell is enclosed by fibrous tissue. How to solve the problem? Well, there are 4 ways. One way is to improve the method and also to improve the polymer material. For instance, the standard method results in sometimes imperfect membrane with exposure of the cell and weakening of the membrane also. We develop new method that can make a perfect membrane with no exposure or weakening of the membrane. Secondly, is to give it orally. We put artificial cell containing CH5 cell that when you give it by mouth can effectively convert hemoglobin, convert red urea into ammonia, which is then taken up by the same cell for protein synthesis and excreted in the feces, effectively lowering urea. And we also start using a more acceptable type of cell from yogurt. And as a result of that, it's now being extensively developed, especially by my PhD graduate, Prakash, into use for different areas extensively, and also by other centers. It works, but FDA does not allow for genetic engineering cell because they are concerned about its use. So, to avoid the use of this, I suggest to my group that we should look at something very safe. For instance, microorganism used in yogurt, lactobacillus. So we work on lactobacillus, show that you can do that. And because of this, it led to development of artificial cell containing related lactobacillus for use, for instance, in kidney failure patients. Kibo originally they want to collaborate with us, but we chose to do it with a Canadian group that did not come up with the funding to do the research. Another way is to remove cholesterol and other things by Prakash doing very well in this work. Another way is to look at regenerative medicine, because regenerative medicine only needs 4 months for liver regeneration. For instance, if you in animal study, if you resect the liver, removing 90% of the liver, if you then put artificial cell containing stem cell, the stem cell remove hepatocyte growth factor that could go through the portal vein into the resected liver at high concentration. That will result in regeneration of the liver. For instance, this is one example of the result we obtained. In rats, we remove 90% of the liver, and the recovery of the rat is shown here. Rats with no liver removed recover no problem and live for the length of the study. Those with 90% liver removed, they die very quickly. Even the free stem cell does not work in this particular case because it doesn't stay in the peritoneal cavity to drain into the portal vein. 90% hepatomy with stem cell with artificial cell result in recovery of the animal. Now, artificial cell containing stem cell could be used, as we just discussed, to increase the recovery of the animal, and indeed the animal recover at the same time the liver also regenerate to the control-free and resect normal animal level as shown here. The mechanism for this could be two. One is the implanted into peritoneal cavity, the hepatic growth factor is drained at high concentration into the portal vein, resulting in liver regeneration. Second mechanism is transdifferentiation into hepatocyte to carry out liver function. But this takes time, and sufficient transdifferentiation only happens in 2 weeks. So this cannot be the recovery of the 42 weeks. Most likely is for long-term possible use. So artificial cell containing stem cell could release hepatic growth hormone into portal vein, result in liver regeneration. However, injecting artificial cell into peritoneal cavity sometimes may cause peritonitis. So for practical reasons, I looked at the other possible thing of asking Dr. Liu to look into transplanting the artificial cell into spleen. This way, it's inside the spleen and function as a hybrid organ. And this way, the hepatic growth hormone is released into the hepatic portal vein and result in liver regeneration. And later, the stem cell transdifferentiate into liver cell and function. The spleen become a hybrid liver organ. Here shows that intraspleen implantation could result in the recovery of the animal and also liver regeneration to the normal animal. The fourth way to do this is to put cell in biodegradable Styrofoam by experiment by Langer from MIT. We won't have time to go into detail. Finally, may I invite you to visit the website. We don't have time to go into any detail. This is just a very brief summary. If you are interested, the detail in articles, videos, and books are available. If you have any questions, I'll be happy to answer, and this is my email address.